Hi, afternoon. It's Monday again. Welcome to day 33 of lockdown learning, and it's good to be back. It was a nice long weekend here. Managed to get away from my desk for a bit, and yes, good to be back today. So let's see where we're starting from to this week. The plan this week is obviously to carry on from where we were last week. So we're going to start looking at the results table that we generated on Thursday, which has got, at the moment, has a lot of rows, has about 60,000 rows, because it's got one row for every gene in our original data set. And what we want to be able to do is to consider those results on their merits, filter those results by various criteria, but also to start generating maybe some visualizations from them, and then think about how to pull in the annotation for the genes, especially the genes that are differentially expressed, but pull the annotation in for those genes to have a look at the identity of the differentially expressed genes. So not just by ensemble gene ID, but by some human readable identifiers. And then I think it was something which I identified on the survey as something that we could do next, which was pathway analysis. But it's kind of the natural next step for differential expression results is to start to think about pathway analysis with those differentially expressed genes. So that, I think, is the logical next step. And I wouldn't necessarily consider that as being separate from RNA sequencing analysis, although, as we'll discuss when we come to it, the source for the gene list that you submit for pathway analysis doesn't necessarily have to be RNA sequencing analysis. So, but it does seem to be the natural follow-on from where we've got to so far. But to begin with, this week we will think about our result table and exactly what we're going to do with that result table immediately. So, okay, with that all said, let's have a look at where we're up to, which is, if you recall, we split our dataset in two because the PCA suggested to us that they were really two separate datasets. And then on Thursday, we analysed the NHBE dataset, so one of the two cell lines, to give us our table of results that we're working with now. So this is the last 20 or so lines of code that we ran last week. So we subsetted our data out. We regenerated our data set and reran the DSeq methods. So estimate size factors, estimate dispersions, and then we ran the statistics over the top of that. And then that gave us our results encapsulated inside our DSeq dataset. And so in order to extract those results, like we extracted the counts using the counts method, to extract the results, we use the results method. And because this is a simple pairwise analysis, results without any other arguments other than our DSeq dataset gives us the results of the comparison between our two conditions, with the first level as the factor as the baseline level. So that's why when we created our factor for our levels, this mock infected factor here, we put the mock as the first level of the factor so that it was the baseline level. That was our comparator level. Okay. So what are we going to do with this result table, the first thing that I need to talk about is what kind of data it is. So, as already mentioned, it's a table, right? So if I run summary result table, it gives me a summary of those results. If I just print out the result table at the console here, like that, then it looks like let's zoom the console so that we can see more of it. Rerun that. It looks like a table. It looks like a data frame. In some regards, at least, because it's got columns, it's got rows, it's got different types of observation in the different columns and rows. It also looks a little bit like a tibble. Because it's got the type of the columns at the top here, whether they're numeric or some other type. But actually, this is not a data.frame or a tibble. It is a, let me just go here. So I'll make a comment. So result table is a data frame with a capital D, capital F, not a data.frame. It's also not a tibble. Now, this is a decision on behalf of the people who wrote DSeq, because they wanted to be able to have things like their summary function, which could do special things like summarize the results of your differential expression tests. And some other things besides, like you will see in a moment, there's plotting functions that work specifically with this results, this set of results. But it's when you're actually working with these results in any meaningful sense to do the kind of things that we do with data frames or with tibbles, it becomes a bit of a pain that it's not a real data frame, that it's this data frame with a capital D, capital F, not a data.frame. So I'm going to do two things. I'm going to keep my result table in its current form, but I'm also going to make a copy of my results table as a data.frame, because for convenience it would be better for me to have it as a recognizable data frame rather than this strange data structure. So first thing I'm going to do is I'm going to make a variable called result.df, which is as.data.frame of my result table. So this is saving my results table as a base R data.frame rather than as a DSeq capital D, capital F data frame, which is a very frustrating thing, and it's a persistent feature of R that lots of people like to implement their own versions of various things, and there's often very good reasons for doing it. But it still can be quite frustrating that there's lots of different implementations of what is basically a data table. Okay, so that's our first thing, and now the view. So whereas last week when I did view with this as data frame inside the brackets, now we can just view result.df. So we can have a look at the results. I've got to actually run that line of code first. There we go. So this is our results object. I'm going to zoom this in, so I'm just going to zoom my source pane again. There we go. So these are the results of our DSeq 2 differential expression test for the difference in gene expression between our two conditions, our mock infected and our SARS-CoV-2 infected NHBE cells. And I want to talk a little bit about this table. So I'm going to take a chunk of time now to talk about the columns and some of the features of this table before we move on to do anything massively significant with it. So our results table has 6 columns, each of which is telling us something about the expression of the gene in each given row. So each row represents a gene, and each column is a set of observations surrounding that gene. So the first being the base mean, that is the average normalized count for that gene across all 6 samples in our experiment. Okay, so that's your base mean is basically the average count for that gene. Where that base mean is 0, so in the case of, for example, ENSG 000005 here, where the base mean is 0, then none of the rest of the columns of the data frame will contain any data, because this gene is simply not expressed. So this gene was removed from consideration for anything to do with that DE-seq does before any test was applied of any description at all. So you'll see that where the base mean is 0, as in this example here, all of the other columns are NA. So there is no other data for this gene. Where the base mean is below the cutoff that was mentioned in the summary, which we looked at on Thursday, but we can see it again if we go back to this console where I printed the summary. Let's just find it. Here we are. You can see that the low counts threshold in my data is for a mean count less than 8. So any rows where the base mean is less than that cutoff, so any rows where the base mean is lower than 8, then those genes are filtered out of the differential expression test. No, actually, that's not true. Sorry, that's not true. They're filtered out of multiple testing correction. So for example, this gene here, ENSG, what's that? 8 zeros and then 938. This gene has a base mean less than 8, so 2.0. So base mean of about 2 here. Maybe 0.2 anyway. And we have stats in almost all of the other columns. So we have a fold change. I'm going to talk about the fold change in more detail shortly. We have a standard error for the fold change, which is quite large, and especially large compared to the fold change. So our test statistic is very small, and our p-value is very large, because this is not a gene which is varying in any significant degree. But because it's got a low base mean, it's been removed by DE-seq before the multiple testing correction has been done. And again, I'm going to talk about multiple testing correction in more detail later as well, which means that it's got an NA for its adjusted p-value. So this has been filtered for low counts, this particular gene. Okay, there's one other category of filtering that I want to talk about, and that's the Cook's distance filtering that I did mention briefly on Thursday. So there's one other class of gene that has been filtered. So you've got the genes which have a base mean of 0, and the genes which have a low base mean. And then finally you have the genes which fail this outlier test according to Cook's distance. And I found one of these this afternoon while I was preparing. So let's just let's find, it was one of the fold change extremes. Here we go, this is one. So this gene at the top here, so ENSG blah 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 265794. Okay, this has a base mean above the threshold for filtering. So the base mean of this gene is 17, which is higher than 8. So this gene is not filtered by that independent filtering, the base mean threshold. It's also got a base mean above 0, so it's not filtered because it's not expressed. But it is filtered. We don't have either a p-value or an adjusted p-value for this particular gene. And that's because this gene has failed the Cook's cutoff filter. And what the Cook's cutoff filter is it's a test for whether a gene has an outlier in its set of observations. And we can it's best to show this by showing you the expression of this gene. And DSeq provides us a method for doing it. So let me go back to my 4 panes and show you this function. So DSeq has a function which is plot counts, which will plot, by default, will plot the normalized counts for your gene of interest. So it takes 3 arguments that we absolutely need. So 3 arguments, the first of which is the DSeq dataset, because that contains the count data and the col data. The second of which is the gene, which you want to plot. So gene equals, and it's the row identifier, so the ensemble gene ID for the gene that you want to plot. And then the int group, so that's the same argument as we had for our PCA plot last week. So int group is the group of interest in your col data, which for us is conditions, same as it was last week. And this will plot a bit like some of the plots that we were making when we were trying to make violin plots of the Golub data a couple of weeks ago. This will plot the normalized count data effectively as a scatter plot in 2 groups, split by that group of interest. So there you go. This is the normalized count data for this particular gene. And what this is showing you is that the normalized counts for 5 of the 6 samples are basically 0. In fact, probably are 0. So you've got normalized counts for 5 of the 6 samples are 0, and then you've got this one sample up here which has a relatively high count in that one sample. And that is the sample that makes this an outlier according to the Cook's distance cutoff. So Cook's distance is calculated for each on the basis of each gene, the Cook's distance for each observation for that gene to the mean expression. And if one of the observations is too far away, then its Cook's distance will be very high and it will fail this filter. And that's what we're seeing for this particular gene, is it just has one single outlier observation. So it's not that the mock samples are showing relatively high expression and the infected samples are showing relatively low expression, and therefore we've got a differentially expressed gene. It's that there's just one outlier which is kind of skewing the whole picture, and so this gene is excluded as an outlier according to that Cook's distance threshold, and therefore, although we have a fold change, a test statistic calculated, we don't have a p-value or an adjusted p-value for this particular gene. So again, this is one which has been filtered by DSeq, and we wouldn't want to consider this in our differential expression results. So at the moment, our dataset has 60,000 rows. We've got 60,000 observations, but not all of those observations are meaningful in terms of differential expression. So it would be beneficial at this point to filter out of this data frame everything which DSeq2 has filtered when it's applied its statistics. it makes sense to remove those filtered genes from our data set before we do anything else significant with it. And so in order to do that, we can remove any row. So if we look at those filtered rows, you will see that any gene which has been filtered has an NA somewhere in the row. Okay, it might be in adjusted p-value, it might be in both p-value and adjusted p-value, it might be in all of the rows in the case where the base mean is 0. So you get all of those NAs. And so those are the things we want to remove. So there's a function in R called complete cases. It's complete.cases, and this will take a data frame and it will return, if every single column has data in it, then complete cases returns true. If any column has no data in it, so has an NA, then complete cases will return false. So this will return a vector of logicals that has the same number of rows as your data frame, and it will be true if every observation is present, and it will be false if any of those observations is NA. And this will enable us to filter out any of those rows which have NAs in them. So we've got our result df. So if I just run this complete cases function, you'll see that it gives me those logicals. It gives me the Boolean values true and false for every row. Where it's true, none of the values are NA. Where it's false, at least one of the observations is NA. Because if you express them numerically, true and false are equivalent to 1 and 0, you can use the function sum to total up all of the ones in there, and that tells you how many rows are true. So there are 15,000 rows that are true in our data frame. So 15,981 rows have all of those observations intact, so have passed all of DESeq2's filters and have produced a result in for our differential expression test. And if we go back to our summary of our result table, I think we can come to the same conclusion by looking at these numbers. So we've got 28,447 with a non-zero read count. So that's a base mean greater than 0. Of those, 12,458 have been filtered due to low counts. So this mean count less than 8 filter. And 8 have been filtered because they failed the Cook's distance threshold. And so we're left with 15,981. This number here is the same as this number here. So this filter, this complete cases thing, is producing the number of trues that we would expect it to produce. And so we can use that because this is a vector of logicals which has the same length as the number of rows in our data frame. We can use it to filter our data frame. So if you remember how to filter using a conditional, one of these relational expressions, okay, we have the name of our data frame, result df, then we open our square brackets, and then before the comma, so we need a comma because it's a two-dimensional data set. It's got rows and it's got columns. We filter on the rows before the comma. We filter on the columns after the comma. We don't want to filter the columns. We only want to filter the rows. So we're going to put our expression before the comma, and that expression has to be either a set of numbers or a set of row names telling it the rows that we want to keep, or a vector of logicals with the same length as the number of rows, where we keep the trues and discard the falses. And that's what complete cases gives us. So complete cases gives us the vector of logicals that we need to filter this data frame. So this will give us back, let's do filter df1. So let's keep everything as we go. So this will give us all of the genes which pass all of our filters, and so therefore we have a complete set of results for. And so that will be a data frame with 15,981 rows and 6 columns. Yes, 6 columns. So let's now talk in a bit more detail about those columns. So let's view filter df1. There we are. So this is our filtered data frame. We've no longer got any NAs in this, so we can talk through each of the columns here in a little bit more detail. So I've already talked about the base mean. So that is the average, the mean of the normalized counts for that gene. So that hopefully is fairly self-explanatory. You can see that these there's quite a range in those values, and that's, bear in mind, that's the average, so it doesn't take into account any difference between any groups or anything like that. It's the average across the whole experiment. The next column is the fold change, so that's the difference between the two conditions in our differential expression test, expressed in as a log2 ratio. And I want to talk a little bit about why we use log to the base 2 to express fold changes. So I'm going to sort of I'm going to pull up an explanation of this that I have. Here we are. So log space fold changes. Let's talk about that for a bit. So it is generally true that in lots of differential expression packages, DSeq not least among them, that fold changes are quoted in log space, so usually in log to the base 2. There are historical reasons for this. So particularly goes back to days of microarrays. So microarrays, the data on microarrays is expressed in log2 space, and so all of the arithmetic is done in log2 space. And Limma, which is a microarray analysis package which is still used extensively for RNA-Seq analysis, expresses RNA-Seq data in log2 scale as well. And so historically, this is the way that you kind of get used to thinking about expression data. There's also practical reasons, and it's the practical reasons that I really want to talk about, and it's to do with the expressivity of numbers. And so let's talk a little bit about that now. Okay, log space data. So let's think first of all a little bit about linear space data. So let's think about the expression of one gene in 2 conditions. So let's think about gene A. There we go. In the boundaries of my, hold on then, let's move this. Gene, that's better. A. Okay, so in condition 1, or let's say condition X and condition Y. Okay, so one gene, 2 conditions. So this is exactly the situation that we're dealing with in our data this afternoon. In condition X, this gene has an average count of 100, and in condition Y, this gene has an average count of 200, and that's after normalization. So this is our comparing. This is what DSeq is doing. It's comparing the expression of gene A in condition X and condition Y. Okay, so in the situation where condition X is the baseline condition, so say condition X is our control, then the expression, the fold change of this gene, I'm trying to get the 200 to appear. There we go. Fourth time lucky. So where gene X is the control, okay, so the way that you then express the fold change of this in linear space is to say Y over X, okay, so 200 over 100, 2 over 1. So the fold change equals 2, okay, so it's twofold regulation, so it's twice as much expression in the experimental condition compared to the control, so it's a twofold change, and that makes lots of sense. And these positive fold changes, so if we think about it on a number line where we have no change being 1, so this is our center point here, so no change is 1, and upregulation we can express using all of the numbers available to us between 1 and infinity. So here we've got twofold upregulation, we would have fourfold upregulation, we could have eightfold upregulation, and so on. Okay, so upregulation, nice and straightforward. If we consider the case where condition Y is the control, so say condition Y is the baseline factor, and condition X is our experimental factor, well then all of this arithmetic changes. Okay, so then our instead of X being the denominator, Y is the denominator, so we're expressing it as X over Y instead of Y over X, which is 100 over 200, which is a half. So that's our fold change, 0.5. Okay, so in linear space then, twofold downregulation is a fold change of 0.5. And what that means, if we extrapolate that from twofold change to these other larger fold changes, means that we have all of the expressivity of all of the numbers between 1 and 0 to express downregulation. So twofold downregulation is 0.5. Fourfold downregulation is 0.25. Eightfold downregulation is 0.125, and we can keep slicing it in half, and you can see that we still have an infinity of numbers, but the expressivity of the number space between 1 and 0 is much more compressed than between 1 and infinity. And so linear space fold changes, they work fine. They do make sense. They make a certain amount of intuitive sense, but there's certain practical problems with expressing our fold changes in linear space. Now, log space goes a long way to relieving these problems. So just to stick with this example, let's think about these fold changes in linear space rather than log space. So, sorry, log space rather than linear space. So let's get rid of my number line for a minute. And let's get rid of my arithmetic in the middle. And let's remind ourselves of a fact about logarithms generally, and that is that the log of A over B is equal to the log of A minus the log of B. Okay. Sorry, I don't know why my erasing of what was there before has not taken It's because Microsoft Blackboard is a bit of a pain. Let me just refresh. Hold on, bear with me 1 second while I do that. Bring that up over the top and refresh while I fix this. So yes, so the log of A over B is the same as the log of A minus the log of B. And so if we want to express our fold changes in log space, we can do it in two ways. Either we can find the log of our expression data, and we should always remember we're talking about log 2 here, log in base 2, the reasons for which hopefully will become clear shortly. We can either find the log to the base 2 of our expression data and then do a subtraction rather than a division, or we can do a division with our expression data and then find the log of the results of that division rather than having to log all of our expression data. So we can work with linear space expression data, so our count data, which is linear space. We can find the fold change, and then we can find the logarithm of that fold change by logging the results of that division. So here we go. My whiteboard is back now with it all being legible. So I don't know what the log 2 of 200 is off the top of my head, or the log 2 of 100 off the top of my head, but I can do the log of the. I can find the log of the results of the fold change arithmetic. So let's go back to condition X being the control. Okay, so condition X is the control. So our fold change then is 200 over 100, which is 2, which means our log fold change, our log fold change is the log 2 of 2, which is 1. Okay. So that's the situation where condition X is the control. If we swap it round again and say, well, condition Y is the control, then we switch the arithmetic around. So it becomes 100 over 200, which is a half. And then that means that the log fold change is the log 2 of 0.5. Let's just log 2 of 0.5. And the log 2 of 0.5 is minus 1. And this is the key thing about logs, which is that the log of the numbers between 0 and 1 are negative, and they're negative with the same magnitude as the positive logs, by which I mean if we draw our number line again, this time our center point is 0. So if there's no change, if the expression of condition X and the expression in condition Y were both 100, then your fold change is 100 over 100, which is 1, and the log to the base anything of 1 is 0. So if you've got no change is represented in log space by 0, which intuitively makes sense. Two-fold up regulation is 1, as we've just found. Two-fold down regulation is minus 1, again, as we've just found by doing the arithmetic. Four-fold up regulation is 2, 8-fold is 3, 16-fold is 4, and so on. And the same in the other direction. So four-fold down regulation is minus 2. 8-fold down regulation is minus 3. 16-fold down regulation is minus 4. And hopefully you can see where I'm going, which is that now we have the expressivity of all of the numbers between 0 and infinity to represent upregulation, and between 0 and minus infinity to represent downregulation. And so suddenly we have this very symmetrical scale for representing fold change, and as long as we understand log maths and how arithmetic in log space works, then what I would hope is that this makes a lot of sense. That each unit, because we're working in log 2, each unit in the log 2 scale is a doubling. So 1 is 2, 2 is 4, 3 is 8, 4 is 16, 5 is 32, and so on ad infinitum. And that's true in both directions. So the magnitude of change is the same. It's just the sign that's different for upregulation and downregulation. So actually there's a lot of practical sense to expressing our fold changes in log space. Okay, so that I realise that's a slightly long-winded explanation, but I always think it's worth explaining that to make sure that we understand what the log2 fold change column represents and why it represents it in that way. Okay, so that fold change has a standard error. So that's the next column, the LFC SE is the standard error of the log fold change, because you can compare if you want to, you don't have to compare the mean of A to the mean of B. You can compare each observation in A to each observation in B, do it pairwise, and get an idea of the spread of the observations around that fold change of comparing the means. And so that idea of the standard error of the fold change gives you an idea of how robust this observation is. So obviously the larger that standard error, the less robust your fold change observation is. And then you've got a test statistic, and this is the results of the world test that I mentioned last week. The world test is a test of fit to the negative binomial distribution. So it uses the mean, that is the base mean, and the dispersion, which we estimated when we ran estimate dispersions. So that those two observations are plugged into the world test and used to generate a test statistic for whether those two populations, your condition A and condition B, in our case, our mocks and our infecteds, are different from one another. So that test statistic is then converted to a p-value, so it's used to derive a p-value for how sure we are that this difference that we're observing, and we always observe some sort of a difference, even if the log fold change is very small. But then the test statistic and the p-value is some measure of whether that difference is significant or not. And the p-value is represented in two ways. That is the raw p-value, which is the output of the raw output of the statistical test. So just, is this gene differentially expressed, yay or nay? And then we have the adjusted p-value. So this is a p-value adjusted for multiple tests, multiple hypothesis tests. And this is essential for this kind of statistics, because we are applying, in this case, we're applying about 16,000 statistical tests in one go. And those statistical tests are not entirely independent of one another. So whether or not gene A is differentially expressed has some dependence on whether gene B is differentially expressed, quite often. So these are linked hypotheses. And therefore, it's very important that we take account of the number of tests that we've done, and we correct for that number of tests. So the standard rubric is that p-value less than 0.05 is sufficient to reject the null hypothesis. That you've got reasonable certainty in rejecting your null hypothesis if your p-value is below 0.05. What that means is that, or in one way of interpreting it, is that if you reject your null hypothesis, you have a 5% chance of being wrong in doing that. And if you're doing a single test, that's fine. But if you're doing, in our case, 16,000 tests, and every single test has a 5% chance of being wrong, then that cumulatively, over those 16,000 tests, leads to an awfully large number of incorrect observations. So in order to control that false positive rate, to make sure that we don't that we aren't making that 5% of our observations aren't wrong, what we want to do is control for that false discovery rate, and we do that by applying a multiple testing correction. So in this case, we apply what's called the Benjamin-Hochberg false discovery rate correction. And so rather than the theory behind it is that rather than each observation having a 5% chance of being incorrect, it's that your positive tests contain a maximum of 5% false positives, which sounds subtle, but it's actually quite a big difference. So if you did 20,000 tests with a p of 0.05, so then 1 in 20 false, you're accepting a 1 in 20 false positive rate essentially. So you make 1,000 false positive observations. Whereas what we're saying is 5% of our list. So if we find 100 differentially expressed genes, then 5 of those will be false positives, which is a big difference. So multiple testing correction in this kind of statistics is really, really important. And so when we're considering which genes are really differentially expressed, it's the p-age, the adjusted p-value column that we're really considering. So, okay, so we filtered our data set according to DSeq's filters, but we also want to apply some filters to the data that we've got remaining, to say not just, okay, well, these are the ones that have been tested, but here are the ones that passed whatever test it is that we want to apply. In order to do that, we need to decide what criteria we want to filter on. What filters do we want to apply? And for an N of 3 experiment, which is what this is, we would normally, so we want to filter the results. We would normally say an adjusted p-value less than 0.05, because we adjust the p-values and derive adjusted p-values, and then we apply the same filter to those that we would do to a raw p-value in a single statistical test. So we're going to filter on an adjusted p-value less than 0.05 and on a log2 fold change greater than 1 or less than -1. So that is a fold change, regardless of the direction, a fold change of 2-fold. So a log fold change of 1 is a linear fold change of 2. Or a log fold change of -1 is a linear fold change of 0.5. So that's 2-fold regulation in either direction. So how do we come up with these filters? Okay, so the first one is relatively straightforward. So filter df1, and we're interested in the adjusted p-value column. So we're using this string notation, so $p-age. This gives me, this bit of code here gives me the adjusted p-value column as a vector. Okay, so that's all of the values in the adjusted p-value column there, and we want to use a relational expression to test whether those p-values are less than 0.05. So that's what we're doing now on this line now, is saying, okay, we want that p-value, that adjusted p-value, to be less than 0.05. This will give us a vector of logicals, which will either be true if that p-value condition is met, or will be false if it isn't. So you can see that there are some trues and some falses in here. And we can filter our data frame by that criteria. So you can see this has 15,981 entries because it's printed 1,000 and it tells us it's got 14,981 left. So this has got this vector has the same length as the number of rows in our data frame. So we can use it for filtering just like we used complete cases up here. Okay, so we do that by saying, okay, so we want filter df2 to equal filter df1. Then we're filtering, so we're opening the square brackets. We're putting our expression, which evaluates to a vector of logicals, before the comma, because we're filtering on rows, and we're not filtering the columns. So this will filter us down to only those rows which have a p-value lower than 0.05. Okay, so filter df2, the dimensions of that, you can see we've got 325 rows in our data frame which have a p-value, an adjusted p-value lower than 0.05. Now, the effect size is important as well, and especially in these cases where we have a small number of replicates. So we only have 3 replicates in each condition. So that's quite a small experiment, not terribly robust experiment. You want to be quite careful about the effect size. So it's not just enough that we've got a significant p-value. We also want to have a large enough effect size for this change to be meaningful with such a small number of replicates. And that's where this twofold regulation comes in. So we want to be able to filter on the basis of the fold change as well as the p-value. So let's find out what those fold changes are. So filter df2, and the column this time is the log2 fold change. And this will give me all of the log2 fold changes in this experiment. And you can see here that there are quite a number that are quite large. So you see at the top, you've got a -3.5. So that's more than 8-fold regulation, because a log fold change of 3 is 8-fold regulation. So that's a big change in gene expression. But some of these, even though these observations have a robust p-value, some of these changes are quite small in terms of the fold change. So down below 1, and down below 0.58 is a 1.5-fold change. So 0.42, that's quite small. Some of these numbers are not huge. And in this kind of experiment with this small number of replicates, we want to remove any of these observations with a fold change less than 1. Okay, so we could filter out the ones with less than 1, but then that's going to filter out everything with a negative fold change. We could filter out everything greater than 1. Everything greater than 1, but that will keep everything above 1, but it will get rid of everything below 1, including all of those with a negative fold change. We could say less than -1, but that would work in the opposite way. It would filter out. It would retain everything with a large negative fold change, but would remove everything between -1 and 0, but also above 0. So everything with a positive fold change. So what we want to do is filter on the magnitude and not the sign. And the way that we do that is by finding the absolute value of the fold change. So the absolute value is the magnitude irrespective of the sign. So the absolute value of -2 is +2, and the absolute value of +2 is still +2. You can find this on a calculator by squaring a number and then finding the square root of the square, because by squaring it you multiply by itself, so even if it was a negative number, it becomes a positive number, and then you find the square root of that to get back to the scale that it was before. In R, we have an easier way of finding this, and that's a function called abs. So the abs function will return the absolute value of all of the numbers. So if we do that with our log2 fold change vector, we will get back the absolute value of all of our fold changes. Be they positive or negative, all of these numbers are expressed as a positive. And then we can filter by saying, well, we want that value to be greater than 1. So we want the magnitude of the fold change to be more than twofold, regardless of the direction of that change. Okay, and that's what this is asking, and will give us a load of falses and a load of trues. And again, we can use this as an expression to filter, like we have done with complete cases and with our test for the adjusted p-value. So this time I'm going to make filter df3, okay, and we're going to filter the previous data frame, so filter df2, and we want to filter it by this expression here. And we want that before the comma, because again, we're filtering on the rows, but we want to keep all of the columns intact. So we're not filtering on the columns. So we have that expression before the comma inside our square brackets to ensure that we're filtering on the rows and we're leaving the columns intact. And so this final object, this filter df3, is a data frame which contains all of the genes which have been tested by DE-Seq, have been found to have an adjusted p-value lower than 0.05, and a fold change of magnitude greater than twofold, regardless of the direction. And let's find out how many there are. Filter df3. Okay, there are 141 genes in total that pass all of those criteria. Okay, which actually, given the PCA, and we look back at the PCA, and we looked at that and we thought, well, that looks like there's quite a dramatic difference between our mocks and our infecteds because they're separated quite widely. The thing that you need to remember is that PCA is based on the top 500 genes by their overall variance. So it's based on the top 500 most variant genes, which is going to be enriched for differentially expressed genes. And also, it's not really quantitative. I know that it contains a percentage of variance explained on the x-axis and the y-axis, but it doesn't tell you how much variance there is in the data. So it doesn't say this is 45% of a whacking great huge amount of variance, and therefore we would expect to find loads of differentially expressed genes because there's loads and loads of variance in this data. They were split on the x-axis by 45% of the variance, but that variance may have been a very small amount of variation. It may be that actually there's not that much difference between the mocks and the infecteds. It just looks like there is in PCA. And PCA is always going to show you the difference, regardless of how small that difference is. It's not a quantitative technique in that regard. And so I expected to find differentially expressed genes, largely because of the expectations given to me by that PCA, but I didn't know how many. So 141 is okay. That's not a bad number. It's quite a manageable number for downstream, for working with these things downstream. So we've got 141 genes. We currently don't know what those genes are, and that's going to be what we're going to do tomorrow, or maybe Wednesday. So I think tomorrow we might look at some visualization. I thought we would have time today, but actually we really haven't had time today. So I think tomorrow we will look at a couple of ways of visualizing our expression results. Certainly, I can think of 2 ways off the top of my head that we will definitely do tomorrow, and a third that we might if we have time. And then I think on Wednesday we will look at annotation. Okay, so we'll do visualization tomorrow, we'll do annotation on Wednesday. Okay, but we have differentially expressed genes, and we can look at those differentially expressed genes. So we can use our view method again. This is a data frame with a relatively small number of observations in, and we can look at those differentially expressed genes, and we can see at a glance that these all fulfill our criteria. That we have our log fold changes are all greater than 1, regardless of the sign, and our p-values are all small. They're all 10 to the minus 2 or smaller. I would suggest if you've got more differentially expressed genes than I have, it's because you've not necessarily applied these filters sequentially. So if you filtered DF1, so if you filtered DF1 on the basis of the p-value to produce DF2, but then you may have also filtered DF1 on the basis of the fold change to produce DF3. So it may be that you've not applied the filters sequentially. So I've filtered result DF to find DF1, then result DF1 to find DF2, and then DF2 to find DF3. But if at any point if you filtered DF1 again here, for instance, then you will have a larger number because you'll only have the ones that meet the fold change threshold rather than the ones that meet the fold change and the p-value threshold, for instance. So that may be one of the reasons why you might find a different number of genes to me. Okay, with that, it's 5 o'clock, so I'm going to call it a day. Thanks for tuning in. Good to see everybody back this week. I hope that was instructive and useful. And tomorrow we'll look at visualizing these results. So we'll look at some common visualizations of differential expression data. Some of them are functions that DataSeq gives us. So like the plot genes, the plot counts that we saw already today. There are functions in DataSeq that allow you to plot out these results, but also I'll show you ways of doing it outside of DataSeq as well, of extracting the data and doing it outside of DataSeq. Okay, so I'll sign off and I'll see you tomorrow. Thanks for tuning in. Come back soon. Keep safe, stay well. See you again. Bye.